this land on which the long COVID web operates. For thousands of years, it has been the traditional land on the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we're grateful to have the opportunity to work on this land. Thank you. Just to recap on the purpose of these webinars, first, it's to share evidence-based information, research results relevant for decision makers, researchers, clinicians, patients and families, and the general public. Secondly, to provide ongoing updates, spotlights on emerging evidence, and scientific information about the activities of our national network. And thirdly, to contribute to reducing and managing misinformation about long COVID. We do have a code of conduct for our network, which is on the link at the bottom of this slide. We value highly engagement around the science and its interpretation, as well as its application. A commitment to reasoned discourse, as stated in our code of conduct, comes with the responsibility for all of us to ensure that any comments or questions remain within the spirit of positive, constructive, and respectful discussion. So we welcome any or all questions and comments in this spirit. Thank you. So just a few logistics. Cameras and mics are all off, but please do post your questions in the chat. We will pause for Q&A after each speaker. We will do our best to review and address as many questions as possible. And just to note that these sessions are recorded, and with permission from the speakers, which we have, they will be posted online following the webinar. Finally, we welcome your thoughts about how these sessions could be most helpful to you. We have a very short feedback survey, which will be posted during the webinar. So just take a minute to complete those. So thank you. I'm going to turn it over to our speakers today. First, we have Dr. Joseph Breen, who's a Senior Scientific Program Director with the NIH Recover Program at the National Institute of Health, Allergy and Infectious Disease. And he will be talking about the current status of the Recover program, including observational cohort studies, pathobiology findings, and clinical trials. Thank you. Over to you, Dr. Breen.
Thank you very much, Susan, and the organizers for inviting me today. So I work in a team across National Institute of Allergy and Infectious Diseases, where I work, and also the National Heart, Lung, and Blood Institute and the National Institute of Neurological Disorders and Stroke, where the Recover program sits really nestled among the missions of the three institutes. So my goal here today is, as Susan outlined, is I want to tell you a little bit about the program overall, and then more importantly, where some of the recent results are in the programs that are ongoing, and then a little bit of forward-looking at the end. So this probably is just background for folks attending this, but in terms of the scope of the problem, and these numbers from in the top here are from our colleagues at the CDC, where approximately 17% of adults age 18 and over have ever experienced post-COVID conditions or long COVID. And at the latest numbers that are available, it's about 7% of adults are currently experiencing symptoms consistent with long COVID. So, and I have a figure actually, and it's from work from Dr. Al-Aly, who you'll hear from later, about where the trajectory of long COVID. And we don't know yet where it's going, but we have a little bit of an idea of where we are today versus where we are earlier in the pandemic. The Recover initiative was launched in 2021 and is really considered an initial investment to understand long COVID. And now I think we appreciate even more the application of long COVID and how its relevance to other infection-associated chronic illnesses. Recover is really has four key scientific aims, and this was set up in 2021. We really needed to understand the clinical spectrum, identify risk factors, and most likely subphenotypes, study pathogenesis over time, which turned out to be very important with the variants, and also the multispectrum disorder, because there are so many different organs that are affected. And of course, very importantly, identify interventions to treat and ultimately prevent PASC. Some of the principles which are involved are the idea that this is a patient-centered program that's national scale, that's inclusive and is really addressing diverse communities, unfortunately, many of which were overrepresented in terms of disease burden for COVID and long COVID. And really to develop platform protocols that then could be tested with a number of different interventions and be adaptive, as now we know very well that the virus has changed over time from the very initial wave. So Recover has, the first really work was really to look at electronic health records as Recover was enrolling 30 different hubs of patients. And in the end, Recover enrolled a full complement of adult and 15 adult cohorts, 2 pregnancy cohorts, and 8 pediatric cohorts, and a number of autopsy cohorts, and combined with those 60 million electronic health records to try and address the aims that I outlined. And data is, of course, really now coming in at a much more rapid clip and increasingly so. So Recover is really designed to be very engaged with patients, with a national community engagement group, and Recover community members interact with, so community members interact with Recover decision makers at all levels. And there were a number of frameworks that were utilized. And some of that's on this slide, using patient groups that were already in existence, and then some were formed on because of Recover. And I think the intention was certainly to get as much input as possible from the research community, the academic community, as well as the lived experience individuals and caregivers. And it's been challenging. And I think moving forward, there is certainly a desire to be as transparent as possible and continue to be informed by the lived experience individuals, caregivers, as well as the academics and clinicians taking care of those folks. And it's been a journey that certainly we expect moving forward, which I'll talk a little bit more at the end, is we hope to continue the engagement and improve it and be more transparent moving forward. So Recover by the numbers, it's a large program. I mentioned the electronic health records, 30,000 adults and children involved in clinical cohorts to understand the disease and continuing to be analyzed. Pathobiology, we're about 2 years into over 40 different projects that were initiated to help understand the pathogenesis of long COVID. There are almost 200 autopsies that have been performed to date. There were over 200 interventions that were evaluated for inclusion in clinical trials, which I'll talk about at the end. And then there are now 8 trials with 13 interventions ongoing. And I'll talk to you a little bit more about that. And again, patients have been involved at all levels here in the protocol design, application review, and are continuing to be involved and engaged. And there have been a number of public seminars which are available on the Recover's website, where folks can go look and see about the scope of the Recover program. There are over 30 of them that are available to date. So a little bit about the clinical characterization from the observational cohorts. So the idea was to understand the clinical spectrum and risk factors, and then really hopefully use some of this information to understand the phenotypes and pathogenesis to really choose interventions that could help. So really better diagnosis and care. So there are some key publications noted on this slide. There were an early look at the adult cohorts in 2023, trying to understand the symptoms. And in fact, some of that data was used to inform the symptom clusters for the first round of clinical trials. And much more recently, the pediatric cohorts, and there's a publication noted there, were done in a similar manner. And as you might expect, the symptom clusters are not the same in the adults. Of course, there's overlap in the pediatrics. There are some insights. Actually, there's a recent paper that was published last week from electronic health cohort looking at reinfection and long COVID and making some interesting observations about the connection between long COVID and what variant one was affected with first relative to subsequent infections. So that's a very recent paper, Hadley in 2024, as well as some distinguishing immune features of long COVID identified at the end of 2023. And I'm going to talk a little bit more about that, some of the things that were noted in that paper in the pathobiology section. So really, this is an indication of some of those 60 papers that I mentioned, trying to understand the cohorts that were enrolled, to understand the disease on a national scale, and then certainly understand the biology involved and to design interventions. So what about pathobiology? Well, there was an analysis published very early on in the pandemic showing some overlap in symptoms that were found in myalgic encephalomyelitis chronic fatigue syndrome, as well as long COVID. And interesting subsequent papers, and meta-analysis reviewing the symptoms, have shown even more overlap between ME/CFS and long COVID, which probably isn't surprising, but actually is hopefully will prove beneficial then for as we move forward with the interventions where the symptoms overlap and where interventions are being tested, and even things like ways to measure outcomes that are more standardized. So there is a great overlap in the neurologic, cardiopulmonary, gastrointestinal, and other symptom clusters. So we're trying to leverage what has been learned on over a great period of time with ME/CFS and long COVID, hopefully to the benefit of both. So this actually figure is from Dr. Ziyad Al-Aly, who you're going to hear from very shortly. And the point of this is they're in their cohort, which he may describe, you could see the symptoms early in the pandemic on the left side compared to on the right side. And the point of this, from my view, is that we are the amount of disease burden is decreasing, but is still substantial. And the thing that this points out to me is that we don't know what the trajectory is. What does this look like in terms of year 4, year 5? Is it going to keep going down, or are we going to reach steady state? So that's really important to understand in terms of the burden of disease, but also what is characterizing that and understanding and addressing it in the neurological, pulmonary, gastrointestinal, all those areas that were identified earlier. So it's really a reminder of we don't yet know what the trajectory is for long COVID. And that's why it's super important that we keep understanding the pathobiology and testing interventions, of course, to try and help those that are suffering. What is the pathogenesis of long COVID? So these ideas came up in the literature. They were noted in that last paper that I highlighted things like viral reactivation of herpes viruses have been proposed, persistent viral infection, which I'll talk about again in a second, and immune dysregulation. Use things like potential autoantibodies or dysregulation of the immune system in general. So the first clue about persistence really came from an autopsy study that was published in 2022 that showed really two major points: that SARS-CoV-2 infects many, many different cell types, which we understand now that we know a little bit about the receptors, but it's still very remarkable and was seen in this early autopsy paper of 44 autopsies. And persistent proteins or RNA were noted in this early paper, but there was no, at that time, no real connection to disease. So it looked like there could be persistent RNA, for example, that could be measured quite far out, up to 230 days after symptom onset, but there was no yet correlation with long COVID. But the opportunity was there that it could be driving disease. So this was an early clue, but subsequently, a number of investigators, and I have one here, one recent paper, asking the question, does persistence play a role in post-acute sequelae of or long COVID? And I think what is clear is that in this study, looking at single molecule array detection in plasma, that there is confirmed SARS-CoV-2 antigen that is present in at least several percentage of post-COVID-19 patients in plasma. Now, how does that link to disease? I think that is the connection that still needs to be made. And there are a number of groups, both inside Recover and outside Recover, working on this finding to try and understand. It's been noted in the literature in multiple places, but it's not 100% clear, particularly the clinical correlation between persistent antigen or RNA and disease or suffering with post-COVID syndromes. The other area that is suggestive is that PET imaging, and these are two representative papers, have seen that there is suggestion for brain inflammation that may correlate with post-COVID syndrome or long COVID. And again, I think this work is continuing and is continuing to be refined by investigators such as those noted here and others to try and understand what could be driving the post-COVID syndromes and of long COVID, but even more importantly, how can we possibly address it with interventions? What about the clinical trials? So Recover developed a number of clinical trials, did a landscape analysis, asked for input from the community, developed the symptoms clusters that were published and I noted earlier, and then launched trials. And they were based on these symptom clusters, really viral persistence and immune dysregulation, based on the fact that we knew a virus, of course, started this, and then neurologic cognitive dysfunction, autonomic dysfunction, sleep disorders, and exercise intolerance and fatigue. Notably, the fatigue has been seen in long COVID as well as the condition I noted earlier, ME/CFS. Eventually, Recover did start 8 separate clinical trials across the 5 platforms, looking at 13 different interventions. And in fact, all of these have now been initiated, and one is actually no longer recruiting the neuro study. So they're in that follow-up period where they're following up patients, and eventually then we'll do the analysis and publish the work. What's next? So one important piece of this is people want access to the data that Recover has generated. And in April, this began to be released in a much more a larger way. The earlier publications did have the data publicly available, as is consistent with NIH policy. But subsequent to that paper, and now Recover is having large data releases on a regular basis through the biodata catalyst, so that researchers can get access to Recover data and really analyze it in a way that they wish to, and not simply as part of Recover's papers, but then can do their own analysis. So in April, 14,000, the data from 14,000 adults was initially made available. And again, these are timed after entry and will continue to be tested. And then the data will be available in the Biodata Catalyst for public analysis. I think I'll stop there, and I'll be happy to answer any questions. And thank you for your attention.
That's terrific. Thank you very much, Dr. Breen. That was an excellent overview of all of the excellent work that's going on in your patch there. So we do have some questions, and we'll run through them for the next several minutes. So the first one, as an executive committee member in Recover, can you comment if Recover will strongly consider recent publication or clinical trial recommendations for long COVID? Dr. Nath of NINDS has made several public pleas for this clinical trial ideas to be utilized in Recover. Any feedback there?
Well, Dr. Nath is well known to us, actually in the MECFS community as well as at NIH. And I think there is Dr. Bertagnolli, the NIH director, has stated publicly that there would be consideration for new trials with some additional funds that were appropriated to Recover at the very end of 2023 or early 2024. And I think that it's anticipated that there will be a way for public input, which would be just that, public input from the academic community, patient community, and include the kind of information that Mr. Hanlon put in his email from Dr. Nath, as well as I believe there might have been some published references in the chat as well. So there should be an opportunity for all of that to be considered when there is a formal process that's open to all for how this is going to be completed. And I think that's what is yet to be really announced and made public. But Dr. Bertagnolli has stated her intention moving forward.
Thank you. I'm not sure if this is related, but do you have any comment with regards to the recommendations to improve Recover from a recent publication or report?
Well, so I think I made a sort of slight reference to that Recover has tried really hard to include lived experience individuals, advocacy organizations, the many constituents and stakeholders. And I think, I think that trying to do that in an even more transparent way, and, it is a goal. And I think since this will be, something additional moving forward, then I think that the intention is certainly to take input and to be as transparent as possible. That's certainly the goal, because, these are public funds. I think that I stated, the burden. And so we want to be as open and transparent as possible.
Great, thank you. From Julie, I would like to know if when recruiting participants in your studies, you differentiate between those who have PEM and those who do not, to be sure not to worsen their condition.
So the last trial that was just started, the Energize trial, specifically does that, actually. If there are two different paths for folks who would want to participate in that trial, if you have PEM at the outset, you would not be part of any cardiovascular therapy, for example, but you would go into the pacing, so-called pacing arm of that trial. So that is sort of the textbook example of how those are being differentiated in that particular study.
And I think the next question from Julie is actually related. So in the analysis, is there a differentiation to separate data from people who have MPE, probably is PEM, from those who don't?
Well, I think we haven't seen the analysis, to be honest. I think that the first trial that finished enrollment is still doing their post-intervention period, and then so we'll have to see. To be honest, I can't really answer that because it hasn't been analyzed yet.
Okay, that's fair enough. From Zoltan, so many organs are affected because long COVID is immune dysfunction related. Is that correct?
So I think there's a lot of evidence for immune dysfunction, which is part of the reason why that first symptom bucket contains immune dysregulation and antiviral uh, ideas for interventions, because that is a commonality. I think it's not as clear in terms of, and I had it on the slide, the role of autoimmunity, for example, in terms of driving disease, but I think that the changes in the immune system that happen with long COVID are certainly noted. So I think that that's a commonality.
Okay, good. And a second part to that, will everyone who had COVID once have long COVID at one point in time? And I guess I'm going to link that to his next question about is there any parallel? Are there parallels to the story with HIV/AIDS trajectory?
Well, so with regard to the first question, it doesn't appear that everyone who gets COVID develops long COVID. That the numbers don't support that. Um, so I think we can't predict the future. It looks from that reinfection paper, it looks like the infection that your first exposure, if it's in a virulent variant, for example, like Delta, definitely can affect that trajectory. So I guess we can't really say down the road, but from the data that's been collected in Recover, as well as the data from the CDC, it doesn't look like all COVID sufferers are going to develop symptoms of long COVID. Um, I guess that's what I can say from that. I'm sorry, there was a second half.
Just about the trajectory in comparison with the HIV/AIDS trajectories. Yeah.
I don't really know what they're referring to. I think where there is a good comparison, I think the HIV folks, because of the changes in the virus, have studied uh, a persistent infection for a long time, and so I think we are trying to leverage the people that have been doing that and the way that that's been done to try and get at this idea of a persistent infection that may be driving some of the long COVID. And I have to say that the other part of that, in terms of working with the community, I think the HIV community is also an example that we can use where lived experience individuals as well as the academic government communities have been able to work well together, and we certainly hope to do that going forward as well.
Thank you. I'm going to allow you to just perhaps use the chat to respond to Billy's question about whether or not there's going to be a meeting in the fall. And then I'll skip because of time just to move to a question from Audrey about how to distinguish between long COVID fatigue from ME/CFS fatigue.
Well, honestly, I think in the future that's going to be right. Up to now, we had this event of infection that was more measurable. I think in the future that's going to be much harder um, to differentiate. And there's a group of people that have had ME/CFS for sometimes decades, unfortunately. And now you have folks who are likely to develop overlapping symptoms that may actually fit criteria for ME/CFS. There's actually Cindy Bateman has studied that and is doing work to try and understand that very question. So hopefully she'll publish some of her observations to help us understand where that's heading.
That's great. Thank you. And finally, from John, are there any plans for NIH to test high-risk, high-reward interventions in the growing area or age of immunotherapy? And a second question there, will there be a larger focus on the pharmaceutical interventions with additional funding similar to what Monica stated in a recent director advisory meeting?
Well, we take our direction from the NIH director, so um, I think I think she's said a little bit about what she hopes going forward, and so I think right now we're putting into place a way for that to happen. And I think that there was a balance of approaches that were chosen in this round, and that was in the past, and I think we'll have to make different choices in the future. And we've certainly heard the feedback and understand why there is a focus and a need for basically drug-based interventions moving forward.
Great, thank you. There's another question there, but I think perhaps if you don't mind, maybe perhaps you could respond in the chat so that we can move to our second speaker. Thank you very much.
Happy to do that. I'll get back to the chat now. Thank you.
And fantastic. Thank you so much. That was terrific. So we'll turn now to Dr. Ziyad Al-Aly, who's Chief of Research and Development at the VA St. Louis Health System. And he's going to tell us about how the epidemiology of long COVID has changed over the course of the pandemic and updates on longitudinal long COVID studies. So over to you, Dr. Al-Aly. Thank you very much for being here.
Well, thank you very much for having me. Can you hear me okay? Can you see my slide? Terrific, thank you. So in terms of outlines today, I want to talk a little bit about how long is long COVID, sort of what sort of long-term trajectories of long COVID, and talk a little bit, discuss a little bit sort of COVID versus flu, our thinking about how does sort of COVID stack up to the flu, briefly talk about mechanisms and offer some reflections and on opportunities and challenges from our perspective. So first of all, how long is long COVID? And again, most of this is really data from my group, so to try to give you a bit of a flavor of what we think about long COVID and what we think about sort of the major areas. So this is a study where we followed people with long COVID for about 2 years, and what we really saw here that there was persistent risk about a third of the sequela, even in the second year after initial infection. So what does that really mean? That really means that in people who got SARS-CoV-2 infection, even 2 years after the infection, they were still coming down with new sequela related to that original disease 2 years ago, and about and those were present, and that risk was heightened or increased in about a third of the sequela that we examined. But it's really important to keep in mind that an infection, even in the remote past, about 2 years ago, can still generate sequela 2 years later. These are the result of the 3-year outcome study. Again, we followed a cohort of people with SARS-CoV-2 infection for year 1, year 2, and year 3. What did we find in the third year? In the third year after infection, we sort of saw it here in this cohort that people were still coming back to the clinic with new onset sequela. These are new problems related to an infection 3 years ago, predominantly in the GI system and in the neurologic system. Again, what does that really mean? That really means that the infection with SARS-CoV-2, people conceptualize it as an acute event with consequences or health effects really immediately shortly after the original infection or initial infection, but that's not really true. That's really misconceptualizing SARS-CoV-2 infection. It can have long-term risks in the sense that, here when we follow these patients for 3 years after the initial infection, they were still coming back to the clinic with new onset health problems in the brain and neurologic sequela and GI problems 3 years after the infection. And this is really important because, I mean, I sort of, the Wall Street Journal, among others, covered it. And then, that mini stroke that you had that you're having now could actually be related to SARS-CoV-2 that you got 3 years ago. And you may have forgotten about that infection 3 years ago, but it didn't forget about you in the sense that it can still wreak havoc in your body 3 years later. So this is sort of an illustration of the burden, specifically here, DALYs or disability adjusted life years, according to different organ systems. Yes, the risk that sort of originates or is manifested anew in the third year is less than that in the first year and the second year, but certainly not negligible. And again, these are measurement of the burden of disease in the third year after infection for new sequela that are happening in the third year. It did not happen in the first year, did not happen in the second year, but then manifested in the third year after infection. So really one of the conclusions from this work that even a remote mild SARS-CoV-2 infection can have health effects 3 years later in the form of incident disease, new disease that was not present in the first year or second year after infection. And this is really a revelation for me because it's like, the wisdom that or sort of the conventional thinking about these infections is that you get an infection, you get fever, maybe respiratory problem, et cetera, and then maybe health effects shortly thereafter. But I couldn't sort of initially, when we got the results, really wrap my head around the idea that these infections with SARS-CoV-2 are actually resulting in new onset sequela, new onset health problems or health effects 3 years later. But now sort of reviewing it in the context of the larger body of evidence that we know about, EBV and other infections, certainly viruses or infections can have latent effect or late onset effects. And SARS-CoV-2 is really one of them. And at least here in the studies that we tracked people for 3 years, and we're still seeing people in the third year after infection manifesting with new onset disease. Very, very, very important. And I wanted to shift my attention and my talk here to a little bit, give it a little bit of characterization of what sort of COVID versus flu. So one of the first 3 matches that we did a COVID versus flu rematch in 2023. And what we saw then, that COVID was leading to 60% higher risk of death than the flu. And then, subsequently, that was in 2023, and then sort of the pandemic was declared, or the public health emergency was declared over. The public was, kept telling us that, well, COVID is not a problem anymore. It's really, it has mellowed down. And we almost ourselves, even here in our team, we almost drank the proverbial Kool-Aid and started thinking that maybe COVID is actually equal to the flu now, until we actually did the 2024 rematch. So even in 2024, even as recently as this, past several months, when we compare COVID versus flu, COVID was still associated with about 35% higher risk of death than the flu. So it certainly is still a much more serious, threat to the health of people around the world than the flu. So what about the long-term effects of SARS-CoV-2 infection or COVID-19 versus flu? We did the studies to sort of evaluate, the outcomes of people hospitalized with SARS-CoV-2 versus people hospitalized with the flu, and we followed them for about 18 months. That's 1 year and a half. What we saw when we surveyed about 94 health outcomes spanning multiple organ systems, the heart, the kidneys, the brain, metabolic problems, GI problems, et cetera, we saw that the risk of complications was higher for about 64 of the outcomes out of 94 outcomes in COVID versus flu. So really COVID, is associated with much higher risk of health effects in multiple organ systems. Here, 64 health outcomes out of 94 health outcomes displayed a higher risk in COVID versus flu. And here is the comparison by organ system. So you can pretty much tell here, if you focus on the the rightmost column here, where we display the red bars, and then at the bottom you see just one blue bar. The red bar represents excess risk in COVID versus flu. What that really means is that COVID is associated with higher risk of heart problems, coagulation problems, fatigue, GI problems, kidney problems, mental health issues, diabetes, musculoskeletal problems, and neurologic manifestations than the flu. The only organ system where flu actually has a higher risk is the respiratory system. So actually, contrary to what it's called, SARS, R in SARS is for respiratory. Contrary what the terminology of SARS-CoV-2 is would suggest, SARS-CoV-2 is a more of a multi-systemic virus, and flu, true to its reputation, is actually more of a respiratory infection. So the flu is more of a respiratory. You see here the blue bar. When you measure the consequences in the respiratory system, flu is actually more severe. But when you measure the health effects or the consequences of the infection in the heart, in the brain, in the kidneys, in the metabolic system, in the GI system, COVID is a much more serious threat to human health. So COVID is more multi-systemic and flu is more respiratory. And this is really very clear in the data. So it's a misconception, actually misnomenclature or naming it incorrectly when we say SARS is severe, acute, respiratory. The R in SARS is for respiratory. That's definitely not true. It's definitely not borne out by the data. If you look empirically what the virus does, this is the R is certainly a misnomer. This is a characterization of DALY, or disability adjusted life years. Similar story here. Sort of this measures the burden of disability and disease associated with the infection. Much more disability and disease as a result of a COVID infection than a flu infection in nearly all organ systems except one, the pulmonary system. So again, flu is more of a pulmonary virus or more of a respiratory infection, but COVID is more of a multi-systemic infection. And here we sort of turned our attention to sort of evaluate, the burden of health or the burden of disability and disease in the acute phase of the infection versus the post-acute phase of the infection. You can really see here the focus on the red and then the blue. So you see more light red than intense red. That's really more burden of disease in the post-acute phase of COVID than the acute phase of COVID. And even in the flu, there is actually more of a light blue than intense or dark blue, meaning that even flu actually gives you or results in a higher risk or higher burden of post-acute events than acute events. That really means that both viruses have actually long-term health effects, and the burden of disability and chronic disease associated with these infections in the long term, in the long phase, eclipses is actually much bigger, much more substantial than the burden of disease that one gets in the acute phase of the infection. When we compared both COVID and the flu together, very clearly, acute COVID is worse than acute flu, and long COVID is worse than the so-called long flu. So COVID is, however you spin it, and even sort of there is a suggestion in the press is now equal to the flu. However you spin it, sort of COVID is a much more serious, remains, even in 2024, despite all the changes of the course of the pandemic, remains a much more serious health problem or problem for health than the flu. We get more COVID cases than flu cases. COVID is year-round, meaning that people can get COVID infection even in the summer. We have a summer wave now in much of the world, including the United States. Flu is actually absolutely seasonal. You don't get flu in the summer. COVID leads to more hospitalization than the flu. That's according to the CDC data. COVID leads to more death than the flu, for sure. Even in 2024, COVID is associated with about 35% higher risk of death than the flu. Both COVID and flu actually have long tails. Infections have long-term health effects, but long COVID is certainly worse in terms of its ability to lead to chronic disease and disability than long flu. Very important. Now, we asked the question, has the risk of PASC changed over the course of the pandemic? Did the risk of long COVID really change over the course of the pandemic? We have a paper addressing this question, which will be coming out tomorrow, July 17 at 5 p.m. Eastern. So for those of you who are on Twitter or X, consider following me. We will put a tutorial out explaining the results and the main key take-home messages from the results. So those will be published tomorrow in a journal at July 17 at 5 p.m. Eastern. So what are the mechanisms of long COVID? So I think Joe touched upon this, briefly. I'd like to, also give you our perspective here. So we did this piece for Science about several months ago. very, very clearly viral persistence, sort of one of the main, key hypotheses here, the idea that some people do not fully clear the virus, that they actually continue to have viral persistence. It may not be the full virus. It could be the full virus, but it doesn't have to be the full virus. It could be viral fragments like viral protein or viral RNA persisting in quote unquote, immune privileged sites that may provoke chronic inflammation and subsequently, chronic disease. Immune dysregulation, and in the sense that the immune system could be either revved up, generating autoantibodies. There is actually now evidence that there is, autoantibodies that even when you take them from people with long COVID and inject them into healthy mice, they reproduce symptomatology of long COVID, suggesting that the that the autoantibodies actually could play a role in the symptomatology of long COVID. And then conversely, some people may have a weaker immune response. They may not even be sufficiently robust to clear the virus. And these two hypotheses could actually be interrelated or correlated in several ways. There is the idea of microbiome dysbiosis, the idea that within us there are actually more bacterial cells than human cells. And when we get sick with SARS-CoV-2 infection, they actually get sick and maybe they don't fully recover. And then that actually may then result in chronic disease or chronic illness. Long COVID or COVID is actually, as we discussed, is not a respiratory illness only. It's actually a multisystemic illness, multisystemic disease, specifically a vascular disease that induces endothelial inflammation, clot formation, red blood cell lysis, platelet activation, complement activation, a whole slew of health problems that can happen in the vasculature, leading to various manifestations throughout the body. And you think like, why is it multisystemic? Why is it all over the place? Guess what? Because you have blood vessels in all your body. They have blood vessels in your brain, in your gut, in the kidneys, in the pancreas, in the GI system, in the heart, in the lungs. Every organ is perfused or has vasculature in it. When the vessel, these blood vessels that actually transport red blood cells and oxygen into those systems, they get sick, you may actually get multisystemic effect. Neuronal inflammation is actually also one of the key hypotheses, and it's actually really quite important because there is a neurotropism, and we'll visit this on the next slide after this, and mitochondrial dysfunction. That is all these sort of the small organelles within each cells that power human life. You can think of them as the battery or the energizer bunnies that actually power our human existence, and they also may get dysfunctional or in the setting of SARS-CoV-2 infection and may not fully recover, subsequently leading to some of the manifestations on long COVID. We think that there's actually quite amongst all the manifestations of long COVID, SARS-CoV-2 could be neurotropic in the sense that it's certainly, we see abundance of neurologic manifestations in the acute phase, in the post-acute, and early in the post-acute phase, and even the risk persists in the nervous system for up to 3 years after infection. And that could be due to, viral persistence, could be due to, dysregulation of cortisol, basically inappropriate levels of cortisol, again, vascular inflammation, microbiome dysbiosis, microglia. These are sort of immune cells in the brain called glia or microglia. They could be activated and then can subsequently lead to disrupted blood-brain barrier. This is sort of the barrier that's supposed to protect our brain from the potentially noxious substances that may circulate in the blood, can shield the brain. Like the brain is our command and control center. It's really, really protected by multiple layers, including our skull and also something called the blood-brain barrier. And then in the setting of an acute infection and subsequently in long COVID, that barrier that protects our brain could be disrupted, subsequently leading to various neurologic manifestations, including brain fog and other cognitive issues. And there is some evidence in organoid, not in vivo, not demonstrated in humans or in living animal models, but in sort of organoids or sort of cell cultures that sort of recapitulate some of the functions of organ systems, that neurons upon infection with SARS-CoV-2 may actually fuse together, and there may be a fusion of glial cells and neuronal cells that may disrupt neuronal activity. Again, this is preliminary evidence and experimental models, not really validated yet in vivo or in actual living animals or in humans. But again, give you an appreciation of the complexity of the biology of long COVID and why we see multiple hits in different organ systems, also including the brain, the brain fog, neurocognitive problems, et cetera, et cetera. So we worry about this problem a lot, and we think that sort of policymakers, when they sort of focus on death and hospitalization from COVID, they're really looking with a myopic eye on the tip of the iceberg and ignoring what's beneath, which arguably is a much more profoundly consequential problem. It can not only lead to disease and disability, but can also reduce life expectancy. We worry a lot about the impact on health systems. When long COVID affect kids or people in school and colleges, we worry about their educational attainment, about their development and educational attainment. It can have profound economic implications. We're starting to see, from multiple different countries, OECD and other reports, really significant economic impact of long COVID. People are not able to, sustain their jobs, not able to work as much as they did before, or even totally abandon the workforce. We worry about sort of global health metrics and how this, the pandemic broadly and long COVID specifically will affect our progress toward sustainable development goals. And we all know, tons of patients with long COVID, and we also worry about their caregivers, about their families. Long COVID does not only affect people, like the patient himself or herself when you see them in clinic, it actually affects their whole family. It affects their friendships, their ability to sustain marriages, to sustain relationships, to sustain friendships. It affects their whole social ecosystem. And it's really a significant disease, and we need to sort of look more broadly. Of course, the patient is at the center and important, but also their family is important. Their caregivers are important. And then they have friendships, and it sort of has really wide ramifications for not only the patient, but more broadly to their, social ecosystem. Here I'll end with a very briefly, because I want to leave some time for Q&A on challenges. We think that offer some reflections on challenges and opportunities. Care of people with long COVID remains suboptimal. We've made some progress over the past several years, but we still feel that people with long COVID are not cared for optimally, not in the United States, and unfortunately not in much of the world. Prevention strategies are underutilized. We know that masking prevents risk of infection. We know that vaccines work. We know that antiviral may reduce the risk of long COVID. We see all these prevention strategies being abandoned, and we know that improving air quality, air purification, ventilation, air purification, or air filtration systems also reduce the risk of infection. But we see them either not being implemented or underutilized. There's no curative treatment. There's actually no validated treatment by randomized controlled trials for long COVID. There remains, despite effort by various agencies and most recently, reports on the definition of long COVID, there's no consensus on term definitions and clinical endpoint. There is really, the field is nascent and embryonic, and there's really no sort of a coalescence around these issues or consensus around these issues. No systems to measure the burden of infection-associated chronic illness. We think that's really actually very important. As you outlined in the discussion following the first half of the talk, there may be long-term risk 5 years, 10 years, 15 years from now. We don't know them. Obviously, we don't have a crystal ball. So we worry about those a lot, because we maybe SARS-CoV-2 still has tricks up its sleeve, and we haven't seen it all. We worry, and one of the challenges that we can is politicization of public health, the spread of misinformation, disinformation, and anti-vaccine and anti-science movements. There is an opportunity here, a lot of opportunities, patient as partners. If there's any silver lining in this whole thing, is that patients actually are leading the way and are doing better than us, I would argue, in helping us sort of understand the condition and lead us to solutions. The scope and the scale of the pandemic isn't a historical opportunity to solve infection-associated chronic illnesses, I think, because we already paid a heavy price for this horror show, the pandemic, and have now, as a result of it, literally, millions of people, actually billions of people who got infected with SARS-CoV-2 in a very short amount of time. So I think we already paid the price for this movement, might as well learn from it. There's an opportunity here to implement better prevention strategies, treatment, data systems and expertise, and preparedness and resilience for the next pandemic. In closing, I wanted to just remind you that SARS-CoV-2 infection can lead to sequelae 3 years after the initial infection. You may have forgotten that you got COVID 3 years ago, but it did not forget about you. It can still wreak havoc in your system 3 years later, even with new sequelae. SARS-CoV-2 is more serious health threat than the flu. You know, again, the flu is mighty. I don't want to come across sort of belittling the gravity of a flu infection, but COVID is worse. COVID leads to more hospitalization than flu. COVID leads to more death than flu. Both COVID and long COVID have long tail, but long COVID is much worse than long flu. Again, thank you for the invitation, but would be delighted to do a Q&A.
Thank you so much, Dr. Al-Aly. That was, as several participants have said, disturbing and amazing, but kudos to you and Dr. Breen for all of the work that you're doing to address these huge challenges. So I'll try to go quickly, and I'll just ask quickly if either or both of you could stay just a few minutes past 2 o'clock just to answer some of these questions. Is that possible, Dr. Breen?
Sure. Just
Okay.
a few minutes will work.
Yeah, we'll try to aim at, say, 3 minutes afterwards. That's terrific. We'll get through as many as we can. So the first question from Rob here, can we be sure that the new onset disease is caused by COVID-19 infection 3 years earlier, or how can we be sure?
So from our perspective, we do these controlled studies. So we have a control group, and then, you know, the risk attributable to SARS-CoV-2 infection or to COVID is really what's in excess of the control group. So these are two groups that are, you know, you can think of as them nearly identical or identical at baseline in terms of age characteristics and other characteristics. One of them is infected with SARS-CoV-2. One group has SARS-CoV-2 infection, one group does not. So the consequences after, you know, in the follow-up period are attributable to SARS-CoV-2. So those are long COVID.
Thank you. When patients do come back to you 3 years after the actual infection, do you measure CD4, CD8 plus numbers? If so, what do you find?
So depending, I mean, I've said once before, like if you've seen one long COVID patient, you've seen one long COVID patient. So depending on which like what it's not a routine measurement. So we don't really, that's not really done as a routine measurement, but it's done depending on if they're in a study, which study they're in, or the protocol of the study, et cetera, et cetera. And again, there is a very broad heterogeneity and presentation and CD counts.
Great, thank you. From Joanne, do the newer 2024 versions of the variants cause as many long COVIDers or people with long COVID as the previous versions of variants like Delta or Omega?
So stay tuned, because that answer is coming out with the study that we're publishing tomorrow at 5 p.m. So we evaluated the risk of long COVID, the risk and burden of long COVID in the pre-Delta era, in the original strain, Delta, and then Omicron, trying to sort of characterize how the risk has changed as the pandemic has progressed. Now, the pandemic is a highly dynamic event characterized not only by the constant evolution of the virus, but also by the introduction of vaccines, which would have also contributed to decline in the risk of COVID of long COVID. So I think that's really that's really the core hypothesis that sort of drove the research question that we addressed in that paper tomorrow at 5 p.m.
Great, thank you.
5 p.m. Eastern.
Mm-hmm. From Audrey, is there any testing for certain biomarkers that can determine if the symptoms or conditions mentioned are definitely due to long COVID? It is mentioned that people with long COVID have distinct patterns of inflammation detectable in the blood, but from my understanding, there's no laboratory test that can identify long COVID at the moment.
That understanding is correct. There's no one single biomarker or one single lab that we can order here. Like we order here, like for example, like, oh, this chem panel or a lipid panel or a hemoglobin A1C. We can diagnose, we can immediately get at diabetes, right? So there's no, like when you get hemoglobin A1C, there is no sort of a single lab parameter, validated lab parameter. Now in the research space, there's quite a bit of investigation to try to evaluate biomarkers, like different biomarkers, and to try to sort of see if there are that we could actually develop diagnostics to be able to diagnose long COVID with biomarkers and also track disease activity that may be helpful for also trials as well. And there's also advanced imaging modalities. You know, there was some really nice work from UCSF where they demonstrated sort of the PET scan, advanced PET scanning with certain radio tracers that you can actually demonstrate continued or persistent, quote unquote, persistent activation of T cells for several years after initial infection that actually sometimes co-localize with areas where there is long COVID symptomatology. So and again, that's really sort of a nascent approach. It's not sort of a validated for clinical use. It's more like a research approach. But there is a lot that has been done in this space, not only in laboratory biomarker, but also imaging, evolving imaging modalities and trying to sort of, hopefully find a signature of disease that can then be used as a biomarker and then validated in clinical setting.
Thank you. From Mary, are you, or perhaps I'll add, are others engaged in any longitudinal studies of ME/CFS similar to what you're doing for long COVID longitudinal studies?
We have not, we have not, but we certainly are. I mean, I mean, the ME/CFS community had actually taught us so much, and the kudos to them, and it's almost like this very big prescience, or sort of a prophetically, they now I discovered that they actually published an op-ed piece about, at the very beginning of the pandemic in the Washington Post, telling us that, as a result of SARS-CoV-2 pandemic, or the COVID-19 pandemic, there may be actually a wave of people with chronic disability because viruses actually sometimes lead to long-term health effects. So there's a lot to learn from them. We ourselves in this group, so we've been intensely focused here, myself and my group, on long COVID, but I definitely, we would be delighted to talk and think with other with other ME/CFS collaborators on how can we help better understand ME/CFS, because we sort of feel that those are all one sort of big entity of infection-associated chronic illnesses, and we need to understand them. And understanding long COVID may help with ME/CFS, and vice versa. Understanding ME/CFS better could also help provide insights into understanding long COVID.
Great, thank you. I notice, Angela, you have your camera on. Is there any questions you would like to highlight or any comments?
Thank you. I just want to thank Dr. Breen and Dr. Al-Aly for great presentations and update on where things are at and what's sort of happening south of the border from us. So thank you very much for being here.
Yeah, thanks so much, everyone. I'm sorry we can't get through to all the questions. They're excellent ideas here, but do stay tuned. We won't be having a webinar in August, but we'll be back in September with more of our series. So just a huge thank you to Drs. Breen and Al-Aly for today. Excellent presentations, and thanks everyone for coming and for your great questions in the audience too. Thanks.
Thank you. Bye-bye.
Lovely summer, everyone.
Bye.
Bye-bye.